I'm going to welcome everybody, but for starters, I want to say that this type of a webinar is set up so that you will not see each other. You will only see the three of us, and you will not have audio either. That being said, though, you will have options for questions and answers. So for this type of a webinar, for those of you who raised your hand, if you could just put your question in the Q&A, and you'll see it's either at the bottom of your screen or it's at the top of the screen, depending on what kind of a computer you have. You'll see a little thing that says Q&A, and Paul is actually going to be monitoring those questions. So if you would just put those in there, I would appreciate it. I see another raised hand.
That was taken off.
Pardon?
They may be writing in the Q&A now. The second
so if you could write in the Q&A, I would appreciate that. You can also put it in chat, but it's much easier for us to for Paul to monitor if you put it in the Q&A. Okay. Let me start. Hopefully that's clear with everybody now. And I want to welcome everyone to Eastman Cares COVID-19 seminar series. We have had several now. As you know, we began back in November. And I am looking forward to this one, and I thank everyone for joining us. The Eastman Cares Education Subcommittee, which is myself and Paul, Elise Kendall, and Sally Wood, and Dr. Ken Dolkart, have put all these together for you. So I want to sort of give them a heads up. I always introduce the Eastman Cares Resource Guide for everyone, which we did update in the fall. It's very helpful. If you want a copy of it, it's both online as well as it's done at South Cove. The other thing I also want to mention again is the eCares Connection. eCares Connection is something relatively new, and it is for people who would like to have a phone call, whether it's a daily phone call, weekly phone call, monthly phone call, whatever. Just if they would like someone to check in and or have someone to talk to. We have volunteers from Eastman Cares who are willing to do that. So if you think you would like to have that, or if you know of someone else who you think might benefit from that, just email us at eastmancares@eastmannh.org. As I said, let's see, we talked about how this is going to happen. Oh, now there's a Q&A, Paul.
Yes.
Okay. This particular seminar is, I'm going to introduce Al shortly, but I did want to mention the two that are coming up. The next one is February 12th, and that's going to be Safe and Effective COVID-19 Vaccines, Light at the End of the Tunnel at Last. That'll be Dr. Liz Talbot, who is our Deputy State Epidemiologist. So that's on February 12th. And then the week after that, right after that, the following Friday, on February 19th, we'll have Mental Health in Times of COVID, and that'll be Chief Justice Broderick. So you'll be getting more information about that as we move forward here. But now, let me introduce Dr. DeMaria. Dr. DeMaria is a good friend of ours. He is currently a medical and laboratory consultant for the Massachusetts Department of Public Health, where he served as medical director for the Bureau of Infectious Diseases and Laboratory Sciences for many years, and also state epidemiologist for Massachusetts. He's a BU and Harvard Medical School graduate, trained in internal medicine at Montefiore in the Bronx, and in infectious disease at Boston City Hospital and the BU School of Medicine. He's a fellow in the IDSA, the Infectious Disease Society of America, and the Society for Healthcare Epidemiology of America, and serves on numerous committees, has written well over 100 publications. He is secretary of the board of directors of the Public Health Museum. I'm not sure if people actually even know there is a public health museum, but there is a wonderful public health museum in Tewksbury, Mass. And if you want more information, they have a website. You can contact me. It is really a wonderful place to take a tour and take a trip if you're interested. Dr. DeMaria has been very involved in that. They have a wonderful course they offer in the summer for high school students called Outbreak. So it really is a fascinating place. And he's currently president of the Massachusetts Infectious Disease Society, past president of the Council of State and Territorial Epis, and the Northeast branch of the American Society for Microbiology. Whew. And he is a wonderful person, a great guy, and both Paul and I have worked for him, and I would say he's the best boss we ever had. And I think you're going to really enjoy his talk. So on that note, I'll turn it over to Dr. DeMaria.
Thank you, Sue. And it's wonderful to join you today. And it's wonderful to see Sue and Paul again. And I think I actually succeeded Paul Etkind as the Secretary of the Board of the Public Health Museum. And what I'm going to talk about today is sort of compare and contrast influenza and COVID-19. And it may overlap with some of the other talks in this series, but I'm going to try to sort of focus on differences and similarities in particular. And hopefully start at the beginning here. Okay. Wait a minute, I didn't share the screen today. Yeah. Is that visible?
Yes.
Okay. So I'm going to compare and contrast, and bear with me because I'm going to start with virology. And I know it's going way down into the weeds, but the fact is, if you understand some of the basic virology of these viruses, it makes everything else much clearer. And these are the viruses under the electron microscope, and a lot of viruses look pretty much the same, and they're basically little balls of protein and lipid fats that surround the genetic material of the virus, and they all have surface molecules, proteins on the surface, and those proteins are very critical because without those, they couldn't infect the cells that they need to infect. And viruses do only one thing. We do many things. We walk, we locomote, we respire, we digest, we have many functions, but viruses only reproduce. That's it. So in order to reproduce, they have to get into a cell and take over the cell's productive mechanisms to make proteins and to replicate their genetic material so that they can go on to infect other cells and survive. And if they can't attach to the cell they need to infect, then they can't do any of that. And this is a little more schematic. And on one side is the influenza virus. That's the one with the blue and red spikes sticking out of it. And then the pink spiky thing is the coronavirus. And you can see the R&D very similar, and many viruses would look the same because they have this motif of the virus particle itself containing the genetic material and the surface proteins. And those surface proteins do various things, but the most important thing they do is attach to the cell to infect. Now, if you look at this schematic of the influenza virus, there's two things I want you to note. One is two of the proteins on the surface are the hemagglutinin and the neuraminidase. So like H1N1, H3N2, you hear these things, and basically those refer to the two protein molecules, the hemagglutinin, which allows the virus to attach to the cell. So it recognizes, in the case of influenza virus, what it recognizes is sialic acid, which is a sugar acid that sits on the surface of cells. It's on all kinds of cells. And when it's presented in a certain way, that hemagglutinin, the H, can recognize it and attach. But because those sialic acids are very sticky, the virus also needs an enzyme to clip that connection and allow it to get off the cell surface and that's the neuraminidase. That's an enzyme, because sialic acid is also neuraminic acid is another name. The other thing about the influenza virus that's unusual for viruses that use RNA for their genetic material is that their genetic material is in segments. So these segments are a little like chromosomes, so they can mix and match, just like the chromosomes of our parents mix and match and result in our genetic makeup. This can happen with influenza viruses as well. The coronavirus, on the other hand, has one long piece of RNA, and that's much more typical of viruses that use RNA. And most viruses use RNA, there are DNA viruses as well. But the coronavirus has an RNA molecule, which is just one long piece of RNA. And it codes for the various proteins of the virus. And in fact, because it's a positive stranded RNA molecule, it's basically messenger RNA that directly codes for protein production. So to make a vaccine, you can snip out some of that RNA that codes for the spike protein. And the spike protein is what attaches to the cells to infect with the coronavirus. So the spike protein is sort of the equivalent of the hemagglutinin. And it recognizes a very specific molecule, a protein on the surface of cells, the ACE2, the angiotensin converting enzyme 2, which is very important in terms of control of blood pressure. Some of you may be on ACE inhibitors or ACE blockers. And these are enzymes that control, to some extent, are involved in the control of blood pressure. But this virus just happens to attach to that on cells. And those molecules are on a variety of different cells. And so you can already see that if you block the attachment of the hemagglutinin in the case of influenza virus and the spike protein in the case of coronaviruses, for example, with an antibody that sits right on there and prevents it to attach to the cells, you can prevent infection. And that's why these vaccines that you're going to hear more about protect you. And in fact, you can also get protection with an antibody against the neuraminidase, the N of HN, to prevent its activity of allowing the virus to detach from the cell. So the viruses find their receptors on the cells. They then use that to get into the cell. They reproduce and they get out of the cell. Sialic acid with influenza virus and ACE2 with the SARS coronaviruses. And these viruses are zoonoses, essentially. They're animal viruses that jump to humans. Now, the influenza virus did it a very long time ago. Coronaviruses, there are 4 endemic coronaviruses that infect us all the time, that cause the common cold symptoms. And these viruses jump from animals at various times in history, as did influenza viruses. In fact, there are 3 types of human influenza virus, A, B, and C. The B and C viruses are only human viruses. So they jumped from their original animal hosts many years ago and became totally human viruses. But the influenza A viruses are basically bird viruses. And they've jumped from birds to a variety of animals over time. And you can see the H's and the N's, certain strains of these viruses. Now, they're not necessarily, so there can be an H1N1 human virus and there can be an H1N1 swine virus, and they can be pretty much restricted to pigs and humans. So the H's and N's don't define the animal host. There can be different H's and N's and different animal viruses, but you can see there's a lot of opportunity there for viruses to jump from species to species. There's a lot of domestic animals involved, and there are waterfowl that are traveling all over the world. So these avian viruses, these A viruses, all of which can infect birds, some of which infect other species, can be widespread. And you can actually have, in a pig, you can have human and avian influenza viruses infect simultaneously, and then you can get those mixing and matching of the strains. Now, you can't get that with coronavirus because it doesn't have segments, but you can certainly, as we only too well, you can get a coronavirus pandemic, because with influenza virus, when those recombinant viruses that have various segments of influenza RNA in them can infect humans, you get a pandemic. And those avian viruses are very prevalent, and you hear about them in the news. H7N9, H5N1 in particular, 15 years ago or so became of great concern as a potential pandemic strain. And we can track each of the pandemic viruses that have occurred in 1918, 1957, 1968, 2009. We can trace the avian influenza virus segments that were involved in those pandemic viruses. And because people can travel from areas of the world where these viruses can emerge, they can travel all over the world. And that's why we get worldwide pandemics very rapidly. But even before people could travel as much as they can travel now, pandemic influenza involved the world because of the characteristics of the influenza virus. It has a 2-day incubation period. In other words, from the time of exposure to the time of symptoms can be as short as 2 days. So every 2 days you can have transmission. Now, with the coronavirus of 2019 coronavirus, that's more like 5 days on average. So you get a little change. In both cases, you can get transmission before symptoms. And in the case of coronavirus, there's a significant amount of symptomless infection that is infectious. And we now know that accounts for a lot of transmission. It can happen to a lesser extent with influenza virus as well, but not really like the coronavirus. And these zoonoses, you remember back with SARS that emerged in China back in 2003, the SARS virus was tracked back first to the masked palm civet cat that was, this is an example in a live animal market that people would buy for food, but then tracked back to bats. And it turns out bats are very critically important in coronaviruses that have the potential for transmission to humans because it can be mediating animals. Just like with the influenza virus, you don't get them directly, generally from the birds, but through intermediary animals with coronaviruses. With influenza virus, the birds are important as a proximate source of an infection. And we're learning more and more about bats and the viruses they can have. Bats obviously live in places like caves in large numbers and have a lot of opportunity for the transmission of viruses in their roosts. So coronaviruses in particular, but coronaviruses, there are coronaviruses and very closely related viruses that are clearly ancestors to coronaviruses that can infect pretty much a wide variety of animals, including invertebrates, as well as even plants. So you can track these back in the genetic record of these viruses and track them back all the way, even to some plant viruses that seem to have a relation to coronaviruses. And coronavirus 2019, the novel coronavirus 2019, is thought to have emerged in a live animal market that was part of this Wuhan market. Maybe, it seems plausible from what happened historically, but just because we recognized it there first doesn't necessarily mean it wasn't already circulating in other areas. And the lesson here is it's happened twice. It can happen more than twice because there are a very wide variety of coronaviruses in bats in other animals, just like there's a wide variety of influenza viruses in a wide variety of birds and other species. And now some people are even talking about pangolins in these markets being a potential bridge animal from bats for the 2019 coronavirus. So you can see from this illustration, the coronavirus here, but bats being the source of SARS virus, as well as SARS-CoV-2 through some intermediary, some bridging animal species, usually a domestic animal, like a pig and like a camel, but also with the potential from animals that are around humans because they're wild animals and they're brought in to areas from the wild to places where they wouldn't be with population densities they wouldn't have. And you can think back to Ebola virus as well as being ultimately a bat virus that was transmitted to other species that probably were the bridge to transmission to humans. And the potential for direct transmission from bats to humans of all of these viruses. And there are some rodent coronaviruses that are thought to have been the ancestral viruses for bovine virus and another virus that infects humans as an ordinary cold virus, HKU1, that may have a source from these rodent viruses. So you can see animals, this is the nature of viral illnesses. It's the nature of human infections that roughly 75% of all human infections have some kind of ancestral connection to animal viruses. In particular, domestic animal viruses. We started living with animals in close connection through animal husbandry. The opportunity for transmission occurs. Now with influenza viruses, I already said we've had multiple pandemics over the last 100, 150 years. And these pandemics have had different characteristics when they've occurred. Many on this call can remember Hong Kong flu in 68. Some can remember Asian flu in 57. It's conceivable, but I doubt it, but people can remember Spanish flu. They can remember the impact of Spanish flu. Many people have family histories that include people who were lost to the Spanish flu, just like 100 years from now, people will have family history of people lost to COVID-19. So the impact of these pandemics has been substantial. And as I said, you can track the development of these pandemic strains to those recombinant gene events in the influenza A viruses. We don't really see pandemics with influenza B or C because they're fully adapted to humans and you don't get that mixing and matching. Now, that mixing and matching can occur, but it would be a human virus emerging from humans and infecting other humans. So it would be difficult to identify that, or it would be difficult for that to cause a pandemic. But the 1918 pandemic has sort of lived in our memory, rekindled memory, I have to say, because it was forgotten for many years, but that had a tremendous impact with about 675,000 US deaths estimated to have occurred. And we may approach 500,000 with COVID-19 before it's under control. So similar number, but remember the population in 1918 was approximately a third of what it was now. So the impact was very great. And in fact, it impacted the estimated life expectancy, primarily because this pandemic is going to have a similar impact in terms of numbers of deaths, but it won't have the same life expectancy as 1918 impact because of the 1918 differentially impacted people between the ages of 15 and 45. And it spread very rapidly. And, you know, COVID virus spread very rapidly. In the 2009 pandemic, H1N1 virus spread very rapidly because that's the nature of influenza. It's very infectious. And when you have a new pandemic virus, it's very infectious in a population that hasn't seen it before. So that's why pandemic influenza spreads so rapidly, spreads so widely, causes so much illness, because it can do it. Remember, in 1918, the fastest you could go essentially was about 35 miles an hour, and within 3 weeks, it was identified across the country. Now we travel much faster, so no surprise, H1N1 and COVID-19 has spread very rapidly across the country with different dynamics in different areas, though. And we need to learn more about why that was the case, but certainly with the potential to cause a lot of disease. And it may be that because COVID-19 has a longer incubation period, possibly is less infectious on an individual basis, or and maybe more important as a super spreader event, that might account for why we saw flare-ups in Massachusetts, Texas, and a few other places, and then flare-ups later on in Florida, Texas, Arizona, and then flare-ups even later in other areas, and then re-flare-ups in certain areas that it's occurred. So we're learning about the epidemiology of COVID-19 as we go along, and I think we're going to learn more as more and more of these viruses are sequenced. And as I said, the 1918 flu was different from any other flu pandemic or any other seasonal flu experience in that it had a differential impact on people in midlife. And so this had a significant impact on people in the working age groups, whereas in all other influenza experience, the impact has been in the very young and very old, but it made us aware that that can happen, and I think contributed to the urgency of planning for a pandemic. And we had very robust plans for a pandemic flu that could have been easily adapted on a national level to COVID-19. And H1N1 affected mainly younger people and had much less of a much less illness and death than with other pandemic viruses. But nonetheless, because it affected younger people and because so many people were infected, it had a significant impact on younger people in terms of the proportion of mortality, because younger people were more exposed, not because the case fatality rate, in other words, among cases, what proportion died was very small, but the attack rate was very large. So the absolute numbers become higher, especially among people who are more susceptible to the complications of influenza. Now, back in the 1890s, there was La Grippe. There was an influenza pandemic that started around 1889, 1890. And this influenza pandemic had a huge impact, especially in areas where it could be observed with urban populations, large population densities, a lot of transmission, and people and the media to report on it. And one of the victims of the flu of the 1890s was Prince Albert Victor, who was the heir to the throne, the son of the Prince of Wales, who became Edward VII, and he died of La Grippe, as it was called, because it really affected France in particular. So his brother became heir to the throne, became George V, wound up marrying Albert Victor's fiancée. Saved the royal family a little trouble because Albert Victor had some issues that related to scandals, whether he was involved or not, still not known. But anyway, it changed the course of the history of the British throne, and it affected the entire world. So it spread over the world, and it was over a number of years. Unlike in pandemic influenza, which tends to go, have a big year, a second wave, a third wave, and then sort of become the common influenza strain. This one seemed to go on much longer than one might have expected with influenza. And of course, they didn't have virology at the time. And there were a lot of other febrile illnesses that were killing people in the 1890s. So it was just one more thing that was causing people to die, including typhoid and a variety of tuberculosis in particular, a lot of febrile illnesses that led to death, and a much smaller proportion of the population was over 60. And interestingly, a number of years ago, I read a paper that I remembered. It looked at the bovine strains of coronavirus and the human OC43 strain of coronavirus. And on a previous slide, I showed you they're closely related. So what they did was they looked at the genome, and you can look at how many differences in the genome between two related strains, and you can track it back in time. It's called the molecular clock, and it was sort of first proposed by Alan Wilson at Berkeley many years ago. And so it's useful in trying to look at the generation of viruses over time and look back at where they might have emerged. And OC43 coronavirus looks like it emerged around 1890. So it raises the possibility that the La Grippe, or the influenza pandemic of the 1890s, was in fact a coronavirus pandemic. And as I said, the world was a different place at the time. So you can imagine exactly the same disease as the coronavirus caused, but in a population with a lot of other febrile illnesses, with a higher overall mortality, age-related mortality rate, and very few people much fewer people over 60. You can imagine it could have occurred and had the same impact as the coronavirus 2019 at the time. So it's an interesting, I think there'll be more research to try to go back and find evidence of this virus. Like they've gone back to 1918 and dug up some people in the permafrost to look at what the virus was that caused the disease. So we've done pandemic planning. Well, I can't remember going back to like 1980 talking about pandemic planning, but it really took off about 15 years ago after avian flu became a concern. And all of the things that we were planning for has happened with coronavirus. Pretty much 40% absenteeism, overwhelming demand, vaccine not available for a while. If you watch the cable news, if they weren't covering the president, they were covering the pandemic 24/7. So very few stories about heroic healthcare workers, but very few stories about heroic health departments. Anyway, we have been planning for that. And both COVID and influenza are transmitted the same way, respiratory droplets. And these respiratory droplets carry the virus. If they get into your airway, your nose, mouth, etc., they can find those attaching receptors and then initiate infection and then spread. So that's what they do. Now, because the receptors of the influenza viruses tend to be on the particular sialic acid receptors, presentations tend to be on airway cells. Influenza pretty much affects the airway. It has some impact that causes fever and muscle aches and all these things that are beyond the airway. But in fact, the infection doesn't really get into the bloodstream or go elsewhere. Whereas the coronavirus ACE2 receptors are in many different cells in the body, in the blood vessels, in the lung, in the heart, in other organs. So that impact of the coronavirus on multiple organ systems is mediated. And again, it shows you how important that receptor is. If you can block that receptor, if you can change that receptor, you can have an impact on the capacity of the virus to reproduce itself. The symptoms, though, even though the coronavirus can infect a lot of other organs, including the kidney, the symptoms are indistinguishable. Larry, there's loss of smell that the coronavirus seems to do because the receptors in the where the olfactory nerve comes into the little sieve in the top of your nose, inside your nose, that allows you to smell, they can infect that. And that seems to be the cause of loss of smell. The fact is, we've tried to make lists so people could differentiate, oh, do I have the flu or do I have the coronavirus? And it didn't work because you couldn't differentiate. You had to do testing to differentiate because they're so similar that it was impossible. Influenza, as I already said, is a respiratory tract disease. There are actually 4 different kinds. There's now a cow influenza virus that is different from A, B, and C. Maybe someday that'll affect humans, but not yet, thankfully. And it can infect any part of the respiratory tract. It's highly infectious. Kids can be vectors, which is different. Again, here's compare and contrast coronavirus. Kids can get coronavirus just like adults can, but for some reason, they don't seem to be important in transmission on a population basis. Whereas with influenza, they're probably the most important factor because you can track influenza in a community. It starts in the toddlers, moves to the school children, then the parents, and then the grandparents. Sometimes directly from the kids to the grandparents. But the children seem to be the vectors of influenza. Does not seem to be the case with coronavirus. An interesting differentiation. Is it real or is it just the way we're looking at it? Because many of these kids could have asymptomatic infection, and little kids can be infectious with influenza virus for a while after they seemingly improve. So we need to learn more about that. Coronavirus, as I said, the incubation is really 5 to 7 days, whereas influenza virus is more like 2 to 3 days, around 2 days, and with the coronavirus around 5 days. But remember, you could be infectious for 2 days before that. So maybe it's not so different if you include the pre-symptomatic period. Asymptomatic infection is probably more like 40% than we know now. And the symptoms are very similar to classic influenza. Classic influenza starts with fever, muscle ache, sore throat coming on all at once when the airway is finally affected by the virus, is damaged by the virus. Coronavirus, when the virus sets up the infection, takes a couple of days, and then the virus starts to reproduce, and then you get the impact in terms of the clinical signs and symptoms of the disease. Most of the disease, like with influenza, is mild to moderate. And this is held up. This is was true at the beginning, and it's true now. It's holding up. 80% of the disease is mild to moderate. And then there's all the people who have the infection and no disease that are asymptomatic. And then the recovery is variable. We're seeing now more and more, because it affects other organ systems, we're seeing the long-term effects that we don't really see with influenza in terms of kidney function, heart function, lung function, et cetera. And we're also seeing a long-term effect of COVID-19, which is being called the long haul, which actually probably does occur with influenza, except we call it chronic fatigue syndrome. So there's probably a great overlap there. And there's something that can happen to you after any viral illness or any infection possibly that disrupts your normal immune system and leads to these chronic effects. That remains to be seen. There's a lot of interesting work getting done on that. Most people have fever, cough, loss of appetite. Some people have loss of smell. I think probably some people get loss of smell with influenza as well, but once you sort of note it, you start noticing it. That's particular, and it may be because of those cells in the nose. And influenza has a big impact every year, seasonal influenza. And let me get to the punchline here. We haven't had seasonal influenza this year. And that's very unusual. And we'll get to that. but every year influenza causes many deaths. Now, they're not direct deaths, it's more direct with COVID, but in a lot of cases with COVID, people are dying of heart disease, people are dying of superimposed pneumonia, people are dying of superimposed complications. So it's not that different, except because COVID-19 affects so many organ systems, you can trigger failure of these organ systems from the COVID infection. And in fact, we used to monitor influenza by death, so-called excess death. So we would look at who died of pneumonia and influenza, and look at what we expected in the black lines, and the red line is what was above the expected death rate that would ordinarily occur if there was an influenza. And every year we go above that a little bit. And you can see back in 2017-18, there was a really bad influenza season. Usually that's associated with the H2N3 strain. In years with the H1N1 that emerged with the 2009 pandemic, we see less. But look what happened in 2019-20. You can see that this was thrown off clearly by the impact of COVID-19, because deaths from pneumonia and influenza, it's hard to tell what triggered them. We don't really know the exact cause. They may or may have been directly caused by the virus, or they may have been caused by complications of the virus, especially in people with pre-existing lung disease. So this seasonal observation of excess deaths we're gonna have to count for COVID-19 in the future. More recently, 20 years ago, 25 years ago, we started looking at influenza-like illness. These are people who present for healthcare who have fever and cough. And they're presenting for everything, but it's the percentage. It's usually like 1 or 2% of people who seek care, even for like injuries, have fever and cough. So that's counted. And it correlates remarkably well with influenza. It's amazing to me that it does, because there are so many other viruses that can cause fever and cough and sore throat, et cetera. But ILI has been wonderful at identifying when influenza is circulating in the population. And you can see multiple influenza seasons here. In the red, you can see we really haven't had one yet, and maybe we won't have one. And it's presumably because all of the things that we're doing for COVID-19 that don't work as well for COVID-19 as they do for influenza is keeping influenza down. Social distancing, masking, etc., is more effective against influenza. Does that mean that influenza is less infectious? Not necessarily, because COVID-19 is a new virus we've never seen before. Whereas we've all been exposed to influenza. Most of us have gotten influenza vaccine. So we have a sort of a baseline of influenza immunity that might be adding on to the protecting effects of the social distancing and masking, et cetera. So we can monitor that. We're learning how to monitor COVID-like illness to try to use the same approach. And we know that ILI works because we do viral surveillance, viral testing surveillance, just like we're doing with COVID-19, and all of the data you're seeing on COVID-19 like this is basically testing data. So confirmed COVID-19 is somebody with a positive COVID-19 nucleic acid PCR test. And we can track it that way, and we track influenza that way, in addition to excess deaths, hospitalizations, influenza-like illness. We can look at COVID-19 deaths. And again, the COVID-19 deaths, it's hard to, if you get COVID-19 and you're recovering and then you have a heart attack, you wouldn't have had that heart attack probably if you hadn't been recovering from COVID-19. So that gets counted as a COVID-19 death. In fact, anybody who dies within 3 months of COVID-19 gets counted as a COVID-19 test. We're gonna have to refine that, because obviously some of those deaths can be taken taken away from COVID responsibility. But on the other hand, there are people who are dying from other things who would have died from COVID-19. So, or that we are dying from COVID-19 without being diagnosed with COVID-19. And then there are excess deaths due to people who are not getting the health care they need because they're afraid of COVID-19 or they don't have access because of COVID-19. So we're trying to sort of sort that all out now. But if you hear criticism about the way we do COVID-19 death ascertainment, be patient because it's in the heat of the pandemic, and we have to go back and learn more about the impact of COVID-19. And this is New England COVID-19 cases, and you can see the states that were particularly affected. As I said before, it varies over the country, and you can see that in the early wave, it was Massachusetts, Connecticut, and Rhode Island. Did I get the colors right? Yes. Unless you're colorblind, you can see that in later, now New Hampshire has come up in the later wave, and Vermont has been relatively spared. Now remember, they have a much lower population density over more of the state than New Hampshire or Massachusetts, or certainly Rhode Island have. So population density is also a factor. And we saw in this fall wave, much more rural involvement because now it took off even in the areas with that lower population density. We also look at hospitalizations by age, and you can see a definite age impact, as you would see with influenza as well with COVID-19. So it's the older you are, the more likely you are to be hospitalized because you have more severe disease, and the more likely you are, especially if you have underlying conditions, comorbidities, to not survive COVID-19. And you can see the incredible racial and ethnic disparities in the hospitalizations and in the deaths. And it's interesting, some studies have demonstrated that if people had equal access to hospitalization and hospital level care, you don't see the racial and ethnic disparities. So it may be a function not only of underlying condition, but also access to care. And really brings home, in addition to many other things, the disparities in health in this country. And the other thing is we can track with hospitalizations underlying conditions, and it's the ones you've heard about. And in particular, unlike influenza, heart disease, lung disease, neurological disease are important, but hypertension isn't that important for influenza, but it is important for COVID-19. And is it a marker for other things, or is it because the receptor molecule, the ACE2 enzyme, the angiotensin converting enzyme, is related to hypertension? So that needs further study. In fact, there was a big concern that people on medications, ACE inhibitors, and ACE receptor drugs, that they may be more, because when you take the receptor blockers, you get more receptors. Maybe there's more receptors for the virus, but it didn't pan out. So there's no evidence that the antihypertensives that target the ACE system make you more prone to, or even less prone to COVID. And then we're finding new ways of monitoring for the virus. In this case, in sewage, we can actually identify the RNA of the virus in the sewage, and it's an early indicator of circulating virus once it gets into the sewage effluent and can be identified there. It comes up even before we start seeing the increases in cases and hospitalizations, et cetera. So that's another thing we're looking at, and we're looking at whether we could do this for influenza as well, because again, hard to distinguish clinically, but we also have rapid tests that are going to be available or are available to test for both of them simultaneously. So prevention, we had a lot of prevention stuff ready for a pandemic. We used it. It was everything we did, we had already planned to do for influenza, except for the masking. And I have to say, we always knew that masking could prevent someone with infection from transmitting it through respiratory droplets. And it's one of our major things we do with tuberculosis, because it's source reduction. If you get rid of the droplets, you can get rid of the source. But there was not evidence base to support everybody wearing masks to prevent acquisition. Now we have that. We absolutely have that. So there have been studies with COVID-19 that shows people who wear a mask are less likely to get infected. And certainly people who are infected and wear a mask are less likely to transmit it. So that's why masks are so important. And it's very sad that we have this anti-mask, non-evidence-based anti-mask and politicizing of mask wearing, because it cost us a lot, I think, in trying to get this under control. Masks do work. We have the evidence for that now. And back in the SARS experience in 2003, in Hong Kong, where they have very good virology, so they could test everybody for a variety of viruses. And when they did everything right, all the other viruses went down. So all the other respiratory viruses that went down, except hepatitis B didn't go down because it's not a respiratory virus, but everything else transmitted by respiratory droplets worked for all of these viruses. And presumably for SARS, but there wasn't enough SARS in the general community to identify that. But the fact is, coronavirus would have expected to go down too. So we have quarantine, we have isolation as well. Quarantine is what you do with people who are exposed, who might become infectious. Isolation is what you do with people who are infected and can transmit. So if you do these things, that's why they're flattening the curve. These graphics were done for pandemic flu, and then they were adapted and adopted to demonstrate what you could do with mitigation. And that's what it's all about. It's about mitigation. That involves things you do about social distancing and masks, and things you do about effective quarantine and isolation to reduce the curve. Because if you don't do that, you're going to see these recurrences. And that's what we've seen in past influenza pandemics as well. We have an influenza vaccine. It's not a live vaccine. It's parts of it's the H's and the N's essentially that works quite well over the short term. But because it's not a live virus, or it doesn't cause a cell-mediated immunity, a memory cell, it needs to be given every year. And because the virus, RNA viruses are very sloppy about how they reproduce. So there's a lot of variation in natural selection. Darwinian natural selection can act on the viruses. When you think about it, the most important way natural selection can act on highly variable RNA viruses is by selecting for more infectious variants, and by selecting for variants that can get around the immune system. And with influenza viruses, getting around that immune system for people who've been vaccinated is why we need to change the vaccine over time, because there's genetic drift. And there's that genetic drift with influenza viruses and with coronaviruses. So they're changing all the time. They have a big cloud of virus. Some of those variants may be more successful than the wild type, the original virus, and they're going to be selected either for their infectiousness, or they're going to be selected for their ability to get around the immune system. And that's what happens with influenza. Now, influenza vaccine on a population basis is quite effective. This was a study done in the UK, where they have one of the reasons why they're so successful at getting so many people vaccinated, because their public health and their healthcare system are integrated, something we'll never have in this country. So they can do a lot of things because everybody has a record in the National Health Service, and it's one system. So what they were able to do is look at everybody over 75 and look at death rates at various levels of influenza in people who are vaccinated and unvaccinated. So in other words, this is deaths from all causes, but you can see the people who are vaccinated, their death rate increased with influenza, but nothing like the people who were unvaccinated. And to me, this is strong evidence that on a population basis, vaccination could be helpful. And then in terms of all this business about I got the flu vaccine and I got the flu, it's, first of all, it's impossible because there's no virus in there. It's impossible with the coronavirus vaccines. You can't get coronavirus from coronavirus vaccines because it only stimulates your response to the spike protein. By the way, it's a totally different spike protein than in the placenta. I've seen some of these anti-vaccine people saying, you know, you're going to get antibodies against the placenta. Well, that's like saying it can change you into Spike Jones because it's totally unrelated proteins. But anyway, this study was 800 healthy young workers. An insurance company said, well, if we vaccinated our workers, could we benefit from that? And in fact, it turns out they saved $4.68 for every dollar they spent on vaccinating their staff. But they also, this is great, they gave half the 400 people the vaccine and 400 people half cc of normal saline. In other words, just salt water. You couldn't tell the difference. The people giving the vaccine didn't know the difference, which was which. The people getting it didn't know which was which. In fact, nobody knew until 6 months after the study, but they called everybody on day 7. And 34% of the people who got the vaccine said, oh, I got the flu. They actually asked for flu symptoms. Oh, I got the flu after the shot. And 35% of the people who got normal saline said they got the flu after the shot. So obviously it has nothing to do with the vaccine. Now, the people with the arm soreness, more people got arm soreness with the vaccine. And I would say that was a good thing. I actually worry if I don't get arm soreness with the vaccine that somehow it's not working. So there's a lot of COVID vaccine under development. There are 2 that have been licensed in the United States. There'll be a couple of more in the next month or so. The RNA viruses vaccines came first because they were easy to manufacture. They're a piece of the RNA of the virus that can, because it's in this little pseudo virus, can get into your cells and then can make the spike protein that then your body sees as a foreign protein. It's only doing that where you inject it essentially. Then you can make antibodies that can block the spike protein and prevent the virus from attaching. Then the next ones are the adenovirus vectors. This is an opportunity for new vaccines because the existing vaccines work so well. There was little reason to commercialize some of these platforms, but they have certain advantages, and it's good to see some of them developing. The adenovirus is a virus that infects cells but doesn't cause disease, and that allows the expression. Now, these vaccines are good in the sense that they give you those memory cells, unlike the influenza vaccine. There's only one live influenza vaccine, and that has to be sprayed up your nose because remember I said it only can infect the airway cells. And so that these RNA vaccines, adenovirus vaccines, and DNA vaccines can give you those memory cells so that potentially you can get boosters in the future if those are necessary, and those memory cells can give you a very robust response to the booster. We also have antivirals for influenza. Now we have an antiviral remdesivir for COVID-19. These work well. They're not exactly like antibiotics working against bacterial infections that are susceptible, but nonetheless, they can prevent hospitalization. They can reduce the likelihood of mortality. And there are a lot of, now this is from April, but I tried to update it. Some of these things have dropped out, but remdesivir and dexamethasone, corticosteroids seem to improve the outcomes of COVID-19. More recently, 2 blockers of IL-6 seem to improve the outcomes of COVID-19. Pretty much everything else is either still in study or has been ruled out. One thing not on here are monoclonal antibodies to the virus. Those are antibodies that are manufactured in cell culture, and those seem to have benefit for people with moderate to severe disease. So that's still under development. There may be more antivirals coming along. One thing I want to cover, I already said wet droplets. It seems that most of the transmission of both influenza and COVID-19 are wet droplets. How much transmission is due to true airborne transmission? Now, true airborne transmission is different. People are using those terms differently. True airborne there's short-term, short-range aerosol transmission, but when we're talking about airborne, we're talking about droplets that actually dry up with infectious particles in them that can still remain infectious, float in the air for a matter of hours, and get on air currents and go to different places. Indoors. Outdoors doesn't count because they're diluted and the sunlight tends to kill them. But anyway, that's true airborne transmission. There's been a controversy for 50 years about true airborne transmission of influenza. And it's come up again with COVID-19, and it's still, there have been no examples of that kind of true airborne transmission, but there's a concern. And that's why there's so much focus on ventilation, et cetera, which is good. There's no question that ventilation is good. There's a researcher at MIT who's doing new studies of respiratory droplets, and what she's discovered is that it's true that the wet droplets only go 3 to 6 feet and then drop by the effects of gravity. But some of them stay airborne as a mist. This is different than those droplet nuclei. They're not staying airborne for hours, but they can stay airborne for minutes. And you can imagine that you could walk through that and pick up the virus that way. And that may explain some of the things that we're seeing. But that's another area of active research. And the other thing is, no two people make the same amount of droplets with the same characteristics, droplets with the same characteristics. It varies. This is in the case of influenza, but it probably already also varies. So that's why there are probably super spreaders who are on one end of this curve that make a huge amount of infectious particles, and then many people who don't make so much in terms of infectious particles. And that might explain some of the differences in some of these outbreaks and things that are being observed. So I hope I didn't go over. I tend to get off on tangents, but thank you. And any questions or comments are very welcome.
So if anyone has a question, this is the opportunity to put it into the Q&A. And while you're typing that in, a couple things. One is obviously to thank Dr. DeMaria for a wonderful talk that we truly enjoyed. Secondly, that last slide was for anyone who really has any suggestions for future.
Oh, should I cut that up? I'm sorry.
Oh, that's okay. Don't worry. I remember it. Evaluations or whatever. Please get in touch with us at Eastman Cares at eastmannh.org. We really value your input. We can't really do evaluations very well with this current format. So we would really like to have your suggestions. We are trying to evaluate, like where do we go after COVID-19, our seminar series, sort of what do we do next and how do we do it and all that. So the committee is trying to make those kinds of decisions now. So we do value your input. The third thing I want to mention is that today is the day, the vaccine sign-up day, to register for your vaccines. We did it, Paul and I did it this morning. And although I just had visions of, the site crashing and all this other stuff. But it really actually was very easy. And you don't get a date or a time. What you will get after when you're done is a notice that says from 3 to 5 days, you'll get an email scheduling you actually for a time. Two things for people who haven't actually done it yet that we learned. One is that when Paul and I did it, I was on one computer and he was on the other, just in case we had trouble. But it turns out that if you're more than one family member, you can do, one person can do it. And when they get through the registration process, they will ask you whether you have another family member, and then you can continue on with that. And the other thing just to keep in mind is they do ask you for insurance information, so you might have to be sure you have your insurance card handy. But it was a very straightforward process. And on that, I see there's a few questions. I'll turn over to Paul.
Okay, first, I want to give you a warm hello from Dorothy Keeney, who's a former Boston school nurse. Saying hello to you, Dr. DeMaria.
Yes, let me, Dorothy, hello. It's so nice that you're here. And Dorothy was one of the
book on the Boston school
great
nurses.
school nurses of City of Boston.
All right, we have a second question.
Who also, let me just say, when you come to the Public Health Museum, as I hope you'll eventually be able to do, you will see an exhibit that honors Annie McKay, the first school nurse in Boston in 1905. She was within 4 months of her starting as the first school nurse, they decided to have school nurses in the entire system.
Okay. How do you perceive the current genetic drift being seen with COVID-19 compared to other coronaviruses?
Yeah, you know, the problem with coronaviruses is nobody paid much attention to them until SARS emerged. And then you know, they've sort of been a backwater in virology with very few people studying them. So the genetic history of coronaviruses is a relatively recent area. And we're clearly seeing genetic drift now, because that's where all the variants are coming from. Because it's not surprising that some of those variants may be more infectious, because again, whatever in the cloud of virus, the variants on the cloud of virus that might be a little more effective in infecting is going to be more successful under the natural selection. So that's important there. And once people are immunized, if the virus continues to circulate, then we're going to have to be concerned about changes in the virus in response to the immune response to the virus, to the vaccine. So that's what happens with influenza viruses. They're being selected for their success and resisting the immune response that you got from the vaccine. And this is true of all viruses. For example, measles. It's a well-known observation that if you get measles from a relative, it's more severe than if you get measles from a stranger. And the sense here is that the measles you're getting from relatives has already adapted to an immune response that is probably more similar to your immune response than a stranger's immune response would be. So the natural selection drives viruses to resist immunity. And we're lucky that if you have good immunity to the measles virus, the virus can't change its receptor recognizing protein in any way that it changes that makes it less likely to attach. So it has a built-in deficiency in that respect.
Okay, we have another question from Dorothy Keeney. Does vitamin D have an impact on coronavirus?
Yeah, there's no evidence of that. There are trials I think are still going on, and unfortunately, vitamin D has been proposed in a number of infectious diseases as being beneficial. And even though there's some evidence that deficiency in vitamin D is associated with a diminished immune response, supplementation of vitamin D so far hasn't been demonstrated to improve outcome with infections. But, you know, we'll see what happens with the vitamin D. I think there's a lot of good reasons to take vitamin D supplementation. You know, especially now, a lot of people aren't going out as much as possible. So even sun exposure, and I think pretty much everybody has their vitamin D level checked, finds that it's low. Yeah. So I think there's good reasons to take it, but I'm not sure that it's going to benefit coronavirus.
And if I can take the privilege of asking my own question, I've been fascinated by the virus, the COVID-19, not COVID, the coronavirus in the sewage, and looking at that as a form of surveillance. Where are we at in terms of correlating what we see in the sewage versus what we see among the people?
Yeah, you know, so far it's very interesting, and a lot of people are doing this now, and they're doing it not only at the large scale level, but even for individual institutions. So in other words, and it's fascinating when you think about it, if you have a prison, you have a college campus, if you could test the effluent, you could see if the virus is there. And it's not limited. I mean, you could identify outbreaks of cryptosporidium. We were talking about this back in 1993, looking for cryptosporidium, cause of diarrheal illness in sewage, because of the whole issue around the water supply in Boston and the concerns after the outbreak in Milwaukee. We actually talked about looking for it, and at the time the technology wasn't really robust enough. But now they're looking for a lot of things in sewage, and what it looks like is that you see it in the sewage before you see people going for care, getting tested, positive tests, hospitalizations, et cetera. So it seems to be an early indicator. That's why there's so much interest to using it for identifiable population groups. Because when you're just using it for all of Eastern Massachusetts, it doesn't, you know, there's nothing to target. But if you knew you didn't have it in the sewage coming out of like a prison, you'd be less likely to need to start testing. But if the moment you start in the sewage and you did testing, you might find it early on in the course of transmission and may have a bigger impact. Likewise on a college campus.
There are no other questions in the Q&A, no hands up among the participants, and nothing in the chat at this point.
Well, thank you again. Thank you, Al.
Stay safe and stay healthy.
And thank you all for attending.
One of my retirement goals was to straighten out. We were going to declutter. We were both going to declutter the house. It hasn't happened yet, despite the pandemic.
Good luck, and thank you again. And everybody go register for your vaccine if you haven't already. All right, bye all.
Take care.